All right, today I'm talking with Steve Shu. By day, Steve is a professor of theoretical physics at Michigan State University, but he also runs a blog called Information Processing, hosts his own podcast called Manifold, has founded several technology startups, including Genomic Prediction and Superfocus, and beyond physics, he's an expert in machine learning and computational genomics. Steve, I've really been looking forward to this one, so welcome.
My pleasure.
Let's get right into your work in genetics. Maybe a good entry point here is with your startup. Do you mind talking a little bit about the background on genomic prediction?
Sure. Genomic Prediction was founded, it must be 5, almost 6 years ago. And our motivation was that on the research side, I and my research group and some collaborators had been working on something called polygenic scores. So I guess it's now called polygenic scores. From our perspective, it was more or less just AI/ML on big data sets, like hundreds of thousands of people, where you have their genome and then you have some phenotype information or disease history information about that individual. And we were trying to do the most basic thing, which is, if you think about like the DNA revolution, what would you like to be able to do with this DNA? What use is this DNA? Well, you'd like to be able to take the DNA of an individual person and predict aspects of that person just based on the DNA, like whether they're high risk for prostate cancer, or whether they're taller than average, or whether they're prone to obesity. So these are all really just fundamental questions that you would be interested in from a basic science perspective. And we had done both theoretical and empirical work, actual work with large data sets. And we're increasingly confident that it was going to be possible to predict to some practical accuracy lots of complex traits describing an individual person. And if you now think about this from the business perspective, like, is there actually any remunerative use of this capability aside from just publishing papers in Nature? What would that be? And the unique thing about IVF is that when a family's going through IVF, it's typical for them to have more embryos. Not every family. I don't want to. Obviously, there are some families that really struggle with this, and they're lucky to get one viable embryo. But it's common, or not uncommon, for families to have many more embryos than they want to use. And at that stage where the embryo is just like 100 cells, there's very little to distinguish two embryos from each other. So what is that family supposed to do? The traditional method of picking which embryo to transfer was to consult the human embryologist who has lots of experience doing this. And by the way, this is a theme if you read my blog about, quote, expert judgment and fake expert judgment. Which if you study data science or machine learning, you immediately realize a lot of times humans think they have signal in their prediction and they don't have any signal in their prediction. So, a typical embryologist would say, oh, embryo 3, that looks like a good one. Embryo 2 is a little rough around the edges. Look, it's asymmetrical. There's a little glob of extra cells on the left there. So let's not use that. And, actually, if you look into the literature, there's essentially zero support, rigorous statistical support for human embryologists in general, or even more specifically, the embryologist at the clinic where you are having any better than chance capability to make that decision for you, right? So you're this is something our critics often forget, is that the thing that we're trying to surpass, the benchmark we're trying to surpass, is random chance, is like no information at all as to which embryo is better than the other. And so what we knew would be the case is that once you could genotype these embryos, the genome predictors or polygenic scores that we had built would give you quite a bit more information about, for example, the future health prospects, risks, tail risks associated with a particular embryo versus one of its brothers or sisters. And so that was the logic, kind of an inexorable logic, that already there's a lot of IVF going on. Already there's a lot of people facing what we call the embryo choice problem. There's essentially zero information, as far as we can tell, being applied, zero real information being applied to solve the embryo choice problem. But polygenic scores and polygenic risk factors and all that stuff would enable us to do much, much better than previously existing methodology. So that's kind of the basis for how the company was founded.
It seems like this work in genomics is at the center of a couple of different disciplines, and we're sort of at this like what seems to be happy coincidence with like multiple different accelerating technologies right now. And so I kind of want to pick this apart one by one. Let's maybe actually start with IVF, because this one to me, like a priori, seems like it would be one of the hardest ones to, shoot up a human female with a bunch of hormones and get like 20 plus eggs or something. But this has been around for like a little while. Can you just describe like a little bit more about how common IVF is and a little bit more about what's going on with that right now?
Yeah. Now, just to be completely clear, because, I'm a scientist and a professor, I don't take any credit for IVF technology that was largely pre-existing before our company was founded. And although we are making actually important contributions now to IVF overall, the basic wet lab technology of how to do that, how to produce embryos and transfer them, is not due to us. I will say that on the board of scientific advisors to Genomic Prediction is one of the scientists who was on the first team that did the very first IVF, produced the very first IVF baby in the UK. So, we have that person on our advisory board. And our patient advocate in the company, Elizabeth Carr, is the first IVF baby born in the United States. So she's the first US IVF baby, and she just celebrated her 40th birthday not that long ago. So it's been around for a while. The basic observation is that by administering a hormone cycle to a woman, you can cause her to overproduce eggs in her cycle, reproductive cycle, and those can be easily harvested. So it's basically a nurse with like a kind of long needle that is able to harvest those eggs, and they're fertilized outside the woman's body. So in vitro in the lab, they're fertilized by a technician, and then you generate potentially a large number of embryos, which then are allowed to grow until they're typically about 100 cells. And by the time we had come onto the market, it was becoming more and more common, certainly at the best clinics, to freeze the embryos once they had reached roughly like 100 cells. And the reason for that is to give the mother's body some time to recover from the hormonal cycle that it just went through before the transfer occurs. And that was found to give better results. Okay, so already the natural cycle is there's going to be a lag period where the embryos are frozen in liquid nitrogen. And by the way, freezing and thawing of embryos seems to not damage them in any way. They seem to work just as well. They're very robust little molecular machines. So basically you freeze them in liquid nitrogen, you thaw them, and generally you might have some loss, but generally it doesn't damage them. Now, it had already, before we entered the IVF scene, become commonplace for a small biopsy of a few cells to be taken from the part of the embryo which is going to become the placenta. So it's not actually the child, but it has cells which have the DNA, same DNA as the child. It's going to become the placenta later on. So the biopsy would be taken from that for genetic screening purposes. So in our startup design, like if you're a startup guy and you're introducing a new technology, you don't want to disrupt the pre-existing workflow of the industry. You want to fit your new innovation in such a way that it doesn't disrupt what had already been there. So that same biopsy, if sent to Genomic Prediction, we would amplify the DNA to the point where we'd be able to get a whole genome genotype for the first time, actually whole genome genotype for each embryo, and then compute all the predictive polygenic scores and stuff like that. So that's a quick sketch for people who are not really familiar with IVF. In terms of utilization, there are some countries where about 10% of all babies born now are coming from IVF, right? So this is a consequence of women having careers, delaying marriage, obtaining more education, just being older when they finally get to the point where they're ready to start a family. And women's fertility starts decaying. Actually, this is kind of, it's just the truth. It's something that people are just not aware of. But women's fertility tends to decay already starting typically in the late 20s, early 30s. So like that might shock you because you're like, wait, my wife is going to be, we have this plan and my wife is going to be 35 when we have our kid. Well, you better look up the statistics because if you're just a little bit unlucky, by the time you're 35, you could have real fertility problems. And almost everybody has fertility challenges by the time you're 40. So, where you are and when your fertility decline begins and accelerates, it obviously depends on the individual. But 30 to 40 is typically where that's happening. So now it's increasingly common for especially like highly educated professional couples to require IVF. And so countries like Denmark, Israel, the state health care system actually supports, will pay for IVF. And so in those countries, it's widely utilized, and the percentage of all births that come via IVF is high. It's like 10%. In the U.S. and other developed countries, it might be like 5% or 3%. But it's a significant number. Like if you're not familiar with IVF, and I just take you to some kindergarten and I point at the kids, like there will be IVF kids among that group of kids on the playground that we're looking at. So it's a non-negligible component of how humans reproduce today.
Okay, and I think that's an important point to kind of send home is this is happening today. People do IVF and you'll be given a set of eggs and you have a choice, which is do you want to pick which one you want to use? And so what you all are doing is giving them options to say, based on some data, we can give you information about what is likely to happen based on which selection you make. So it'd be interesting to get a little bit into how you all make those predictions. There's kind of like two components, right? There's actually gathering the data. So we need a bunch of genotypes with labeled phenotypes for different traits. And then we also need the machine learning models to like actually do the AWS run or whatever, right? So let's maybe talk about the data first. I'm really curious. I noticed that like in your papers and others, like the UK Biobank is referenced a lot. Can you talk a little bit about like where data exists today and like where most people are getting it to do research?
Yes. So most of the data that's available for researchers like us, to do our work, comes from government-funded large biobanks. So UK Biobank is probably the best known one, but there's also one, more than one, funded by the NIH. There's one called All of Us, which is approaching, I think, well, it's not quite at 1 million, but the target is to get to like 1 million individuals. There's something called the Million Veterans Project, which is run by the healthcare arm of the Veterans Administration, which is also around 1 million people. My lab, in particular, also collaborates with the Taiwan Biobank. And so in Taiwan, we have available something like 1 million genotyped individuals to analyze. There's a biobank in Finland. There's a biobank in, there's one in Japan. So it's all over the place, really. However, it is true that most scientists in this field spend a lot of time trying to get access to data. A lot of what we're doing is data limited, not algorithm limited. So I would say like the innovations developed by my group in terms of ML algorithms to do this kind of work, they're already good enough actually to work quite well. And the main thing we're waiting for is actually just to get more data. And you might say, oh, well, wait, don't you have millions of people that you can analyze? Yes, but you need to break it down to, like, say you're studying a particular disease. What you really need to get signal is you need to compare the genotypes of cases. So cases in medical research means individuals with the disease condition, and controls, so people who don't have the disease condition. And if the incidence of the disease is 1% or 5%, suddenly the number of cases is much smaller than the total size of your biobank, right? And most of your signal is really coming from the cases. And in the case, for example, if you're studying cognitive ability, it's very hard to get cognitive scores, for example, for individuals in the sample. So it's a complicated field. It's now a big field. Like when we published our first paper on height in 2017, where we successfully predicted height with pretty good accuracy, a few centimeter accuracy, which was a shock to the research community. From that point, where we were one of only a few groups really focused on polygenic scores. Now, I think each year there must be, I'm guessing, like order 1,000-ish papers published every year, which are doing something with polygenic scores. So now it's become a big international area of research.
Yeah, let's dig in actually to the height thing, because this seemed to me like actually a really big deal. So tell me if I'm getting this right. Was it that you all predicted how much data ahead of time you would need to accurately predict height? And then you got that data and you were able to accurately predict it? Is that roughly how this went? Because that seems like a really big deal and could have implications for other traits moving forward as well.
Correct. So I won't get the years exactly right, but I'll get them roughly right. So I think in 2014-ish, around 2014, maybe it was a little bit earlier than that, we published some theoretical papers. So at that time, there were no big biobanks available for analysis, but there were smaller data sets. And we did some theoretical work, and there's a bunch of fancy math here that goes under the name compressed sensing or L1 penalized regression. There are even papers, very well-known papers by this, in which one of the co-authors is this Fields medalist, Terry Tao, on this subject. So there's a purely mathematical question of, if I give you noisy data of a certain kind and you use a particular set of algorithms, how much data do you need to solve the problem, to recover the signal fully? Okay, so that's a signal processing problem, or you could think of it as an information theory problem, whatever you want, problem in analysis. But it's pretty widely studied, mostly in the applied math community, a little bit in electrical engineering and computer science, much less so in genomics, even computational genomics. So very few people in the field of computational genomics are aware of these compressed sensing theorems, which I was aware of. And actually, in deciding to work in this field, so that was about when I decided to work in this field, being a theoretical physicist, I said, I'm not just going to randomly choose some area to start working in. I need to understand theoretically what is possible. If I'm going to invest some years of my effort in this direction, I want some theoretical guidance that, it could turn out like you need hundreds of millions of genotypes to get anywhere. And if that's true, when I do this back of the envelope calculation, it's going to happen for a long time, 20, 30 years. So I'm not interesting to me, right? If the answer had come out like that, I would have been like, let some biologists slave over this, whatever. But so I did that preparatory theoretical work. That is common in physics. So in physics, theory is very highly developed. So before you do anything, you do preparatory theoretical work to understand the problem, and then you decide, is this worth doing? Is it worth building this accelerator? Is it worth putting the satellite in space to look at the Big Bang? We need to understand a bit more about the problem before we invest resources in it. Biomedicine is completely the opposite. Biomedicine is just like wild-ass speculations about stuff, blowing huge sums of money. NIH has more money than all the other sciences and engineering research budgets combined. It's just because senators understand dying of cancer, but they don't understand anything else. So basically, NIH gets all the money, and then biomedicine is just like throwing money in the air like this, and they don't have any theoretical understanding of what they're doing. They're just trying stuff. Okay, they'll reject what I'm saying, but this is what any theoretical physicist or mathematician or computer science who gets involved in biology will tell you. Is it like they're actually in a way like not respected, like they wouldn't respect a mathematician, because generally math is not that useful for them. Like math doesn't help them with what they're doing. They have to do this very, experiment intensive, empirically driven stuff. That's just how their field is, because living systems are so complex. But so what I'm saying is, I think, not politically correct, but it's easily verifiably true. But for my own effort, I said, let me do this theoretical analysis first to figure out whether solving the genome, in other words, predicting phenotype from genotype, is that a solvable problem with realistic amounts of data and compute that I'm going to have available to me? Or should I just give it a skip and continue thinking about black holes and quantum fields? So I went through that. We published our papers, I think by 2012 or 2014, we had published the results. which is using some very fancy math, something called the Donahoe-Tanner phase transition in compressed sensing. We were able to use that to calculate and predict how much data it would take to solve a complex trait, using realistic genomic data. And so we predicted that as soon as we had at least a few 100,000 genotyped individuals for whom we had height measurements, we would be able to build a reasonably good height predictor. And the very first moment when a data set of that kind became available was 2017, when the UK Biobank released its first instances and allowed researchers to apply for access. So within a month of getting access to UK Biobank data, we had built predictors that had this few centimeter accuracy. It was shocking to the genomics community. If you look at the journal Genetics, where we published the paper, Genetics is the preeminent genetics publication in the United States. So it's the journal of the American Society of Human Genetics. So it's the leading journal in the U.S. which is specifically about genetics. Our article is an editor's starred article or something. They have some way of putting a gold star on an article that means the editor thinks it's an important advance in the field. So that's an editor's selected article in the fall. I think it was I forgot when it was. I think we posted the preprint in the fall of 2017, and the paper was published in early 2018. It's an editor's selected article, and they even I have the referee report, so you can see the referees saying "Wow, I can't believe this is possible," right? So people now, younger researchers who maybe entered the field since then, or maybe they were already just starting grad school back then, they will say, and maybe they're even being sincere, but they don't know the history of their own field. They will claim that none of this is surprising. That we always knew we were going to be able to do this and stuff like this. It's all bullshit, because if you were in the field in 2017, people were saying oh wow, there's all this missing heritability. I don't know how we're ever going to solve this missing heritability problem. I think all genes interact with all other genes, so therefore modeling complex traits will never be possible. And if you're a serious historian of science, you can go back and analyze what were people saying in 2015, 2016, 2017, up until the point our paper came out. You can see they thought what we were doing was impossible. I mean, it's easy for people to verify this. Now they will deny it, because now polygenic scores everything. It's a big deal. It's a big research area, and they'll claim, "Oh, we knew this all along," whatever. But this is like insider baseball for how science works. There's a joke that says scientific discoveries the reaction to scientific discoveries go like this. First reaction, it's wrong. You can't do that. What are you talking about, Steve? All genes interact with all other genes. You'll never be able to predict this. It's highly nonlinear. So number one, it's wrong. And then the next reaction is, okay, maybe you're right, but it's trivial. I knew it all along, right? I knew it all along. Of course you did. But you did it, but it's not that big a step. We all knew it was possible. So it's wrong, it's trivial. And then the final one is, I did it first. The person says, you were wrong, and then, your result is trivial, is now saying, I did it first, please reference my papers. So that's a common cycle for acceptance of new scientific insights. And I'm not the first person to formulate it that way. Even people Heisenberg and Planck and other people said the same thing. Quantum mechanics, they said, it's wrong. Oh, okay, you guys might be right, but this is kind of trivial. And then oh wait, I did it first. I knew it was going to work like that. So anyway, so we predicted how much data we would need. When that amount of data became available, we solved the problem. And now it's been, our result was replicated by several other groups, prominent groups. And now it's even been replicated more thoroughly by a study which used, I think, several million individuals for whom they had height. And so now it's basically incontrovertible that a trait as complex as human height, for which you need to know the state of about 10,000 individual regions of your genome. So there are about 10,000 different genetic variants that are used by the ultimate predictor in order to calculate the estimated height. So it's 10,000 individual loci with different effect sizes. And now, again, in 2017 or 2015, people would have said it is insane to think that you crazy data science, AI, ML guys will be able to build something that complicated that depends on, think about biologists, right? Like how many biologists can count to 10,000, right? So they would say, like, it's insane. You're never going to, genes all interact with each other. You guys will never be able to build a predictor that uses 10,000 different genetic variants scattered over all chromosomes that impact this really complicated trait. Okay, but now it's been fully validated. Anybody who knows about computational genomics agrees. Height is, there are very good height predictors. They use about 10,000 steps. And furthermore, even though I just stated, like, okay, it's impossibly complex from the viewpoint of a wet lab biologist, from the viewpoint of information theory, from a computer science viewpoint, it's incredibly simple because there are 3 billion different variants that could have affected height. 3 billion. So 10,000 out of 3 billion is a very low fraction of all regions of DNA that actually matter. So actually, as we predicted, height would turn out to be a sparse trait. All traits, actually, all complex traits of humans are sparse. They only depend on a small fraction of the 3 billion variants that exist in the genome. So, and furthermore, the effects are largely additive. It's a largely linear model that accounts for this. So it's not even this weirdly nonlinear thing that these guys have been hallucinating about. So anyway, in a way, like, height is the poster child for what can be done in complex trait genomics. It's pretty well understood, at least the people who are experts in this field. I think the understanding of that is not propagated that far outside of, like, computational biologists who are really computational genomicists who are really actually secretly kind of computer scientists and statisticians, to, like, old school genetics scientists who, like, think their whole career about, like, one gene or something. They don't use a lot of math. That group doesn't really understand what I just told you. And then going out to other biologists who don't deal with DNA data on a regular basis, they have no idea what I just told you. So anyway, so that's my history in this field.
I can remember back in I was in grade school in the 2000s, but there was a big deal about the Human Genome Project, right? And there was like Dolly the clone sheep, and it seems like there was all this optimism about the field, and then it kind of went dark for like the next 10 or 20 years. Everyone got excited about the iPhone or whatever. But now this stuff is so recent, and what has been shocking me as a layman is exactly what you said. Intuitively, I would think that these polygenic traits are just like impossibly complicated. And maybe if you reduce the risk of like diabetes, you're like jacking up cancer or heart disease or something. But you guys have also shown that like typically that is not true. You can even create like a generalized health score. So in some cases, there's even like mild optimistic effects for other health measures. Is that right as well?
Yeah, so it's a great point that you're raising. And I just want to point out that someone who approaches this subject with kind of an open mind, and I think your background is in engineering and software development, if you can, if you have decent math chops and a scientific technical background, and you just approach this area with an open mind, you can read papers written by our group or other groups in the field and understand, like, what is being said here now about genetic architecture? Because up till now, like, although we could read out genomes, we didn't really know what they did, right? That was the Human Genome Project that when you were a kid, around 2000, they sequenced the first human genome. And those guys then, as always with all like biotech entrepreneurs and tech entrepreneurs, like overhyping what's happening, right? So they're like, that was just the ability to read out an individual DNA. The analysis by which you figure out what can I predict based on that took this next 20 years, basically, right? An accumulation of large numbers of genomes, right? So another fairy tale that was invented by biologists when they literally knew nothing about the first genome had not been sequenced. They knew nothing about machine learning. They knew nothing about compressed sensing, etc. there was this weird fairy tale invented by them called pleiotropy. Okay. And pleiotropy says like a particular gene or a particular locus in your genome is bound to have multiple effects, right? Exactly why that had to be true. Okay. You could say it this way. A gene, by definition, is a region of DNA that codes for a protein. And there are only about 20,000 different genes in the human genome. But of course, between each gene, there are tons of other DNA switches that are doing other things, which people until recently had no idea what was going on with these other things. Of course, from an information theoretic standpoint, you realize like it can't just be the information in the protein coding regions. There had to be these other switches that are controlling things. Otherwise, like how could I be that different from a worm, even though we have pretty much the same, we're using the same proteins, slightly different variations of these proteins, me and the worm, right? So, but we're very different, right? So there must be other information that's involved in making me versus making the worm. So I think like, to be totally blunt, a high IQ kid like who's 12 years old who reads a popular book about DNA would understand this, but it's still today confusing to most biologists. So there was this feeling that, okay, because these genes are everything. So for a long time, these guys thought genes are everything and this is junk DNA. Okay, genes are everything, but there are only 20,000 genes. So therefore, if I modify one gene, I have to affect lots of different things, because for sure that protein is playing a role in like lots of different things going on in your body. Hence the idea of pleiotropy. Okay, so now once you have predictors, that we have predictors for 20, 30, 40 different complex traits, ranging from diabetes risk, to schizophrenia risk, to height, to BMI. You can just look at the predictors. And they're sparse. Remember, they use only a fraction of all the information that's on your genome. Each one of them is only using a fraction. Now, the fraction is scattered all over the genome, on your chromosomes, but it's not using most of the SNPs, most of the genetic variants on your genome. So you can just ask this trivial question. You can say, like, well, how many of the SNPs used for height are common to the predictor built for schizophrenia? And how many of those are common to the predictor built for heart disease? And how many of those are, common to the one that's, predicting your diabetes risk, right? So, or whether you have brown hair, okay? And someone, again, who knows a little linear algebra, but most biologists don't know any linear algebra, would say like, oh, why not compute a correlation matrix between these predictors? Like, look at the correlation between variance accounted for between these different predictors, and let's see how disjoint, using a fancy mathematical term, let's see how disjoint are the input SNPs, the sets of input SNPs for each of these different phenotypes, different traits. How disjoint are they? Are they very disjoint, in which case pleiotropy is bullshit? Or are they hopelessly not disjoint and overlapping, in which case pleiotropy is correct? Turns out they're largely disjoint. Okay? So now, how many biologists actually understand this today? Very, very few. You have to understand something about like information theory, or maybe at least linear algebra, correlation matrices, and polygenic risk predictors, or trait predictors. And then combining that, it's trivial to then, oh, gee, that old term in the textbook I had in graduate school called pleiotropy, whatever happened to that? Then like, oh, if you understand these 3 or 4 different conceptual areas, you can just immediately answer the question. Okay? But again, hasn't propagated very far. Now, another way to say this is the following. So it's just like basic math that like when 2 physicists talk, we're always talking like this kind of math. But again, like not everybody can do this. So if I say to another, if I'm like trying to explain all these results to some guy who's, his day job is, studying, black holes in anti-de Sitter space or something. And he's like, oh, Steve, I hear you've been dabbling in this biology stuff. Why are you doing that? And, and then like, well, what did you guys find out? And I'm explaining all this. And then the guy will immediately ask me, he'll say, well, between any two humans, how many individual variations are there? Like how many, if like my genome and your genome, how many differences are there? The answer is it's on the order of a few million out of 3 billion base pairs. So it's about 1 per 1,000. So there are millions of ways in which my genome is different from yours, right? And if the most complex trait that we found so far, which is height, uses 10,000 of those variations, if I divide a few million, let's suppose the average predictor, polygenic predictor, is using a few thousand genetic variants, okay? So let's suppose I divide a few million by a few thousand. The answer is 1,000. Okay, so physicists are always doing these order of magnitude calculations, right? So it turns out there's enough information between any pair of two humans to specify about 1,000 different complex traits if those complex traits were all independent of each other. So it's a trivial information theory calculation that says the common variation found, differences between two randomly selected humans, is about enough to account for 1,000 completely independent complex traits, which are roughly like the complex traits which we have studied so far. So that means if you're playing Dungeons and Dragons, okay, and normally in Dungeons and Dragons, your strength is independent from your dex, which is independent from your intelligence, which is independent from your wisdom, which is independent from charisma. I forgot if there are more. Constitution, maybe it's 6. Okay. Characters in D&D live in six independent dimensions, right? It looks like humans, given the amount of information in our genomes and the amount that we vary from each other, could live in 1,000-dimensional space of complex traits, even assuming exactly zero pleiotropy, where each set is entirely disjointed from the other. So I don't know if I went through that too fast, but it's kind of trivial for some people logic that says, hey, wait a minute, yeah, there might be some pleiotropy, but actually there are a lot of independent ways that I can tune people's genomes and get what I want. Like the size of my spleen is actually not directly related to my IQ. And the length of this finger could be somewhat independent of the length of this finger. You know, like there are 1,000 different things, ballpark, that could be basically not interfering with each other, or interfering only weakly with each other, right? So that's the situation. Which, if you look at papers we wrote, like 2020 and 2021 or something, all this is laid out in those papers, but not understood by a lot of people. By the way, part of the problem is that to get money from NIH, you have to focus in on one narrow thing, maybe one disease or something. So then, people who know the diabetes polygenic risk, index, they know that, but they know nothing about heart disease. They know nothing about height. They would never study height. They would not study IQ. They don't even know what IQ is. So of course, if you're a jeweler with these magnifying glasses, you're looking only at one part of this watch. Yeah, you don't know what the weather is outside. Okay, but that is kind of how effort is specialized in biomedical sciences.
So this seems like, I keep using the word, but happy coincidences. It's kind of a miracle, right? It's oh, wow, there's actually room for potentially a lot of important traits that we can just maximize with potentially no downside. So I don't know if you're a sci-fi person and you get excited. Maybe some people are kind of repulsed by this, but if you get excited about it and think, hey, it could improve human outcomes in the future, I'm curious, let's put the societal barriers and constraints aside. So things like data collection and general ethics of, choosing between embryos and things like this. But from a purely technical point of view, what do you think are the biggest barriers to us in the future being able to have people that have substantially better health outcomes and they're more fit, they're better looking, they're smarter? Do you see any real potential technical gotchas, or are we looking at mostly an issue of ethics and data?
I think you have the right perspective on this, that in a way things turned out in the most positive way, because it could have been that the encoding of genomes is so complicated we would never decipher it, or we would need 100 times more data than we have today in order to decipher even much simpler traits. That could have been the case. That was the main thing I was worried about. I was mainly worried about nonlinear interactions, making the code space much more complicated than it turned out to be. Okay, that was my main worry in 2015 or something like that. But then once we got the data and started working with it, I realized, it ain't like that. But so I gave an interview to the Sunday Times a while back, and the title of it was something like, "Yes, superhumans are possible." Because like that's what the science is telling us. Now again, like most people are not aware that like the science is strongly pointing toward the answer being yes, superhumans are possible. But I'm telling you, yes, superhumans are possible. So if you look at one of the things we did in my lab is we created an overall health index. So we took genomic predictors for like 20 different disease risks, the major disease risks that kill people. We took about 20 of them. And we computed an index, we created an index summing over each of the risks for those major disease conditions and then weighted by the life expectancy reduction typically associated with those conditions. So the most impactful has the biggest coefficient and the least impactful has the smallest coefficient. You just sum it up and you make a health index. And similar things have been studied by other groups too, like the Finns also studied this. They have a nice study of their Finnish population gene bank, also using a similar longevity index. And it looks like to us, because again, the individual SNPs that are controlling health risk 1, like heart disease versus diabetes, heart, number 2, lung cancer, all these things, most of those sets of SNPs are disjoint. So it looks to us like we could, just using what we already know, like the information that's in the predictors as it is, you could hypothesize a human who's a low-risk outlier for each of those 20. So when we genotype people or embryos, we can just compute this health score, right? And so we can see like, oh, Johnny is low risk for this, medium risk for this. Poor Johnny, he's a high-risk outlier for high blood pressure. So we can do that. Now, there's no reason, as far as we can tell, judging by the specific genetic variants, the individual SNPs that are involved, there's no reason there cannot be a person who's bottom, 1 percentile risk, so low risk for each of these 20 conditions. And who knows how long that person would live? That person might live 150 years, or, the number of people who have been simultaneously low risk for all of these conditions that have ever lived might be zero. Like, there have been enough humans around to realize the luck involved in being a 1% positive outlier in terms of low risk for all of these killer conditions. Maybe that person has never lived. Okay, maybe it's 0.01 to the 20th chance of this happening or something, right? So, but you could get there in principle by engineering or by having a huge number of embryos or whatever to choose from. So as far as we can tell, this particular phenotype, longevity or disease risk, that's not what people are afraid of. People are not afraid for us to talk about this. You could even get funding, from NIH to talk about like longevity, risk predictor, whatever. The ones people don't like are ones like intelligence. So the question is like, oh, well, if I do this trick with intelligence, I look at the predictor, that the predictors, genetic predictors for intelligence are not that strong yet, but you can at least see kind of some trends. And it does look, there's a lot of variation up for grabs there, and you could shift the mean. You could move an individual many standard deviations if you wanted to. Like if you, in principle, if you had perfectly accurate editing capability, how far could you shift an individual in their IQ score? And the answer is you could shift them really far, probably beyond like any human genius, that historical genius that has ever lived. That particular analysis, all of a sudden now, if you're kind of like a left-wing extremist, whatever, or woke person, you would say, oh my God, you're talking about shifting intelligence. You must be a eugenicist. Like you must secretly salute the Nazi flag in your basement or something. So that one's a little more fraught. But the math is basically the same math that we were talking about when we were talking about longevity.
So let's talk about intelligence, because I think this is where we've been sort of building up to this, but I think this is where it gets really, really interesting. One of these technologies that we need, other than the data and the training runs, is we also need the ability to either pick from a bunch of embryos, which is going to be like a generation by generation sort of improvement, or we can edit an existing embryo. You mentioned it in that response, but we haven't talked much about editing. What does the current state of editing look like?
Yeah, so of course, editing a human embryo is a big no-no. Like, a certain guy spent a few years in jail after doing this in China. So obviously, the first thing we should say is like, well, this is not considered an ethically okay thing to do. We're just talking about science fiction. You and I just happen to be interested in the character Khan from Star Trek. You're too young to remember this, but anyway. Okay, we're having a science fiction discussion. It's about Dune. Like in Dune, they were trying to breed Kwisatz Haderach. Okay, that's in the novel Dune. So we're talking about science fiction now. Currently, there's been continued improvement in CRISPR technology. The thing that you're contemplating, though, is a situation where you might make hundreds of edits to an embryo. So there are hundreds of different places where you figure out, okay, I want to make a change here, and then you do it. And currently, the technology, as far as I know, does not exist to do that without also having significant risk of off-target edits. Okay, so edits that were unintended by you. Okay, so that there's a wet lab gene editing molecular biological limitation right now to our technology that makes this hard. The other limitation, which is a little bit subtle, is that when we build these predictors, we're using a particular SNP of the state of your genome at a particular place in order to help us make the prediction. But we don't know that that particular SNP that we're using in the predictor is itself causal. So the state of nearby SNPs, because they tend to be inherited together, are correlated. And so we might be using a tagged SNP in order to do the prediction. We only need correlations to the prediction. So we might be using a tagged SNP to do the prediction, but the causal SNP is actually next to it. And if we edit the tagged SNP, but not the causal SNP, we get no effect. So the more difficult problem, which is more of an information theoretic or computational problem, is to determine not just which SNPs are enough for me to predict a phenotype, but which ones are actually causal because I actually want to change the phenotype. So that is also an unsolved problem. And that problem will require a lot more data than we currently have because of this problem that it's hard to tell if in almost all the people in our gene bank, the state of this SNP is the same as the state of these two states are correlated like at 0.9. It's very hard for me to tell whether this is the causal one or this is the causal one. And you have little clusters of these things in your genome. So there's a kind of technical problem at the computation level that is a roadblock to gene editing supermen.
But okay, so gene editing, it sounds like there's still some work to be done. But what about if we just iterated over a bunch of IVF generations? I think there was a paper done on this actually that tried to figure out like how many standard deviations you could get out of intelligence.
Well, it depends on a lot of parameters, like how many embryos you have to choose from, and then also how good is your predictor for making the selection. These calculations are pretty straightforward. Like, I mean, once you make the model assumptions, you can do the calculations. The problem is we don't know the model parameters because we don't know how good our predictor for intelligence will be in 2030 or 2035, et cetera. Like there are a lot of unknown parameters that would have to feed into the calculation.
How good do you think it'll be for intelligence if we had just straight IQ tests, like the best IQ tests that we have today?
Yes. If you had well phenotyped individuals, so they had been given like, even just the SAT or some very good IQ test, cognitive test, and you had, my estimate is if you had, let's say, a few million people with such phenotypes attached, you could then build an IQ predictor with, accuracy maybe plus or minus 10 IQ points. And so from that, then, you could start selecting, you could have, every generation. Well, it also depends on how many IVF resources you give to people. Like if it becomes a social convention for women to freeze a bunch of eggs, do extraction cycles when they're 20, and freeze, the resulting, like maybe they could get 100 eggs and freeze them and then use them later in life. So you're selecting the best out of 100, and you have a pretty good predictor. You could be moving the mean in the population like 1 standard deviation every generation. So that would be huge, because in a few generations you would have an unrecognizable human population. Like you'd have, the whole population of the planet is like the student body at MIT or something, right? So
Yeah.
But of course this is all hypothetical science fiction and possibly morally wrong.
Just based on like the human species, how many standard deviations is it possible to go? To push this intuition a little bit, if you think of like, if we just engineered dogs, presumably there would be a limit and we wouldn't have dogs that could, maybe they could get close to us today, but like presumably they won't have the same limit that like humans do. Is there a way to think about that?
It's pretty hard to do it from a mathematical perspective. So what we can conclude by the fact that the level of polygenicity of cognitive ability is probably at least as great as height. So it's probably at least 10,000 different variants that are controlling the common variation in cognitive ability. And there's a little math involved in this next inference, but it turns out it's the square root of that number that determines how many standard deviations are up for grabs. So if you take the square root of 10,000, let's say, very conservatively, you could say there are at least 30, maybe 100 standard deviations up for grabs. So you could shift the mean. Now again, like a standard deviation is 15 IQ points. So if you shift 30 standard deviations, the IQ went up by 30 times 15, which is 450 IQ points. And we have no idea what the hell that means, right? Because we're used to thinking about variations of 10, 15, 20, 30 IQ points, not 400 IQ points. So the inference that you can make is that there's an unimaginable amount of variation up for grabs. If you think like, that's crazy, Steve, some other limiting factors are going to intercede before you get plus 100 or plus 200 IQ points. Might be true. But our experience in agricultural breeding, where the similar analysis applies, okay, so if you look at a plant, by the way, this whole field that I'm talking about, which again, like I'm always like taking the piss from these stupid wokesters, is like, they don't like me talking about this stuff, but like if they go down to the Monsanto lab or the Iowa State University Agricultural Breeding Center, people are doing essentially the same mathematics with plants and animals in an agricultural setting, right? And oops, I guess it's not BS. So in the agricultural setting, they have a similar situation where like the milk production of a cow or the number of eggs laid per month by a chicken, or the rate of growth of corn, or the size of the ear of the corn, or the drought resistance of the corn, they can tell by the same analysis I just gave you for IQ, they can tell that there are many, many standard deviations up for grabs, and if they aggressively start breeding these plants, or using polygenic scores for selection, which by the way now has become completely standard in like breeding of dairy cattle and stuff like this, and I think maybe even for some cases like breeding chickens and stuff like this. There are many, many cases of multi standard deviation shifts that have been actually accomplished by animal breeders and plant breeders. So the eggs that you ate for breakfast are laid by chickens that lay almost 1 egg per day. So in the wild, they might lay like 1 egg per month, or maybe a couple eggs per month. But these chickens are laying an egg almost every day. So the chickens that are populating all of our farms, they're ravenously hungry. They just want to eat and lay eggs. They are, in the wild population, less than 1 in a million, 1 in a billion wild chickens from the old population were anything like the modal chicken on a farm today. So that tells you right away, like stuff that's unimaginable, like when you look at the wild guy, can be produced through controlled genetics. And this is just true. And if you ask any animal science guy, people who do animal breeding, plant breeding, they'll just tell you they'll just tell you, yes, of course, that's how we have agricultural revolutions, and that's why farms are so productive today, and yada, 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 yada. So I think it's very unlikely that, as a purely scientific statement, what I'm saying about what's possible with human intelligence, I think it's very unlikely that I'm wrong about this. Now, it could be like, oh, the humans that are plus 100, IQ points or whatever, they start to have bigger brains, and their skulls have to be bigger. Brain size is correlated at about 0.4 with IQ. Other things could happen that you don't like, right? Like, oh, maybe all women would have to have cesarean sections in the future because the baby's brains are getting so big because you did this weird genetic editing to them, right? And I'm not endorsing any of this, but the fact that there are many standard deviations up for grabs, I think, is, for people who actually understand the science, is not arguable.
Just so people understand, a 130 IQ, right? As you said, that's like 2 standard deviations. So that's like you're in the top 2.5% of the population.
Right.
A 145 score, you're in the top 0.14%. You're getting into 160 to 200 is like Einstein, speculative on some of the smartest people of all time.
Yep.
And what this is showing is, using the same logic we do with the chickens that we eat today in my kitchen, you could theoretically push this to get somebody that's up in the 500s or higher.
Yeah, now I don't know what it means to have an IQ of 500 or this or that, but I know that person probably is a hell of a lot smarter than me. Now, the other funny aspect of this conversation, it's like, if you can tell, like, I'm kind of sick of talking to like biologists and stuff, or biomedical people. Like, yes, it's true. I spend most of my time these days working on things more related to AGI and AI and large language models, stuff like that. And there, like, if you say like, oh, you know what, we're going to 10x the training data and 10x the compute resources, and I have a slightly better algorithm, am I going to get an AI which is significantly better, smarter than GPT-4? People are like, yeah, wait, Steve, that's what we're doing. What are you talking about? Oh, you were in biologist mode or something. You couldn't understand things for a while. Like, why would that be shocking to you? Like, why would it, like, right now, GPT is not good at certain things, but the guys who are working on this are very confident that we're going to pass some threshold, and suddenly the new models will be magically really good at some of these things, like theory of mind, or doing mathematics, or whatever it is. That's not shocking to them. Like, for people who think very deeply about intelligence, the operation of an information processing neural network, which, by the way, my brain is one, right? And your brain is one. So people who think about that stuff don't think it's crazy to talk about something that is a quantum leap in capability beyond the previous one due to some improvements. Well, could those improvements be encoded by genetic changes in the organism? Why not? Why the hell not? Like, why is it that my dog will never understand doorknobs, but my kid will understand the doorknob by the time he's 2 or 3 years old or something, right? And wow, it must be God or something, the Bible, or like what could possibly explain that? Oh, I guess it has something to do with DNA, except we shouldn't talk about that for reasons or something. But yeah, it's kind of ludicrous. So the idea, like, there are biologists who, like, they'll read some like popular article about like genetically engineering super geniuses, and then they'll give you like 5 fallacious reasons why it's impossible that we'll ever genetically engineer super. They'll like put their own stupidity on like public display by giving you like 5 fallacious reasons that have nothing to do with the problem, and which, if you map their reasoning onto what people are doing training neural networks, it's just obvious what they're saying is stupid, right? But anyway.
I mean, you could stretch this analogy because it like kind of plays into the whole, like, concerns about AGI. It's like, well, who's going to get smarter faster?
Oh, this is the main question. So I think for the most sort of deep thinking minds in our society today, the main question is, at what point will machinic intelligences overtake human intelligence is one question. And second question, is there any hope for the wet intelligences to improve themselves to affect that race? So the race between them, we're not getting better. We're actually getting worse if you calculate carefully. And then like they're getting better, right? But the fact that we could get better at a slightly different, at an accelerated rate through these biological technologies, the average people don't want to talk about this, but serious people are thinking about stuff like this. So yeah, that's a question of our age, actually.
It's also almost like symbiotic too, because some of the hardest problems of creating smart intelligence are human intelligence to build the thing to hit escape velocity. So you get smarter humans, they could, be the ones that are now pushing the machines a little bit further.
A conversation that I regularly have with, billionaires who are very focused on AI is whether improved humans, like if we suddenly were able to make much smarter humans, would have a better chance of solving the alignment problem so that the risk of the machinic intelligences doing bad things to us is reduced. And, should we try to slow down AI developments so that improvements in human intelligence can, have a better shot at solving the alignment problem? Not solving it, but improving engineering around the alignment problem. These are serious conversations people have. Again, it sounds like science fiction, but serious people are actually having these conversations.
I mean, it's here. And like, maybe this is a good segue into, we kind of laid the groundwork for like what is possible, but we glossed over some of the like societal challenges to actually getting here. Yep. So one thing I wanted to ask you about is, when I think of like the really groundbreaking stuff, it seems like you have kind of like nuclear on one end, and we've proven to be like pretty good at regulating it. It's like very physical. It's like not easy to get your hands on plutonium if you like don't know what you're doing or have special authorization. And then you have AI on the other, which is the jury's still out, right? It's like once you do a training run, you can like gate the thing behind an API, but unless you're confiscating like H100s or something, like it's very difficult to actually understand like how you're going to stop people from like doing these big runs. Yep. So this seems to kind of fit somewhere in the middle, right? And I think at one time I did 23andMe like quite a while ago, like when it was first coming out. You could just download your raw data, yes. And there are some sketchy sites that will like tell you things that 23andMe either doesn't want to for reputational reasons, or it might not even be real science, but they can like try to tell you more about your genome than 23andMe will give you. So the question here is basically like, once someone does one of these runs, and let's say they get their hands on the intelligence data and they do it, like what is to stop me with my 23andMe or like an embryo that like could be my child from me just like sending it to them and saying like, hey, can you go like pick the best one? Is this something that's going to be really challenging actually in the long term for anyone to stop from happening?
So I think the main difference when you compare these genetic technologies to either in silico AI or to nuclear weapons is, the barrier to entry to do the genomic stuff is actually low, other than the difficult part of assembling the large data sets. That's hard. But once you have the large data sets, the computational analysis and the IVF stuff, it's kind of low CapEx stuff in comparison to the other two. Now, the part that's slow, though, about genetics is it proceeds only at a human generational timescale. So you have a lot of time. Like if you made some mistake and you screwed something up for this generation, you have a lot of time to study them and like fix it. So like, I think the nightmare scenarios about runaway human genetic engineering or whatever are actually way overblown. Like there might be a particular family that suffers, or even part of a generation of people that suffers, but the human species is not going to like destroy itself by genetically engineering itself. I don't think that's a huge risk. More risky is their ability to genetic engineer viruses and things like that. That's a whole different thing. But I don't think there's going to be any stopping the genetic technologies that we're talking about, because even if the United States is taken over by wokesters and by the way, just as a historical footnote for your listeners, if you want to have some fun, go look up a guy called Lysenko in the Soviet Union. So under Stalin, the study of genetics was actually forbidden, and lots of scientists were thrown into the gulag for studying genetics because it violated socialistic tenets of the equivalence of individual men and stuff like this, right? So anyway, long story, but the point is, in America we're kind of like flirting with ending up in that place, but it's not going to stop in China or Taiwan or Japan. Like other people are going to eventually get millions of IQ scores or college entrance exam scores linked to genomes. And so yes, eventually there will be predictors. These predictors can easily run on your phone or on very limited hardware, right? So eventually this cat cannot be kept in the bag. Even if it is kept in the bag by some risk-averse, woke, Americans or Europeans, it's not going to be kept in the bag in Asia. No way. So there's no holding that back, but it's going to play out over multi-generational timescales. And so like my AI friends, it's like I have different groups of friends, right? I have the physics friends, I have the AI friends, I have the computational genomic friends. It's like when the AI friends look over at the computational genomic friends, they're like, don't those guys realize like everything they're doing is going to be like replicated by some AIs like in 20 years, like in 5 seconds? And like, so and like who cares how smart humans are, because by that time machines are going to be like 100 times smarter, right? So in a way, like from that perspective, the issue is like, well, the machines are going to surpass us, and then at some point the future of the planet will be in the hands of the machinic intelligences, not the ape brains.
I wonder too, if you just surveyed the United States, like how many people would actually care about optimizing for intelligence? Like, what if instead we just get like everyone looks like Brad Pitt and Angelina Jolie, or like celebrities,
Oh, you're totally right. Many more people care about that than care about brain power. I mean, the smart kid was never the most popular kid in school. And it's not even very easy for people who are kind of more in the middle of the distribution to even understand how the people in the tails drive progress. Because by definition, if you're in the middle of the distribution, you don't know how a large language model works. You don't know how a thermonuclear bomb works. You don't know how your microwave oven works. You don't know how your internal combustion engine works. So you don't even know that, well, wow, if I did a really careful accounting, I'd notice like predominantly it's these weird smart kids that we didn't like in high school who are responsible for all of those things that I just listed, each of which has like totally radically changed society. But these people don't know it or care about it. Much more immediate to them is like, is Johnny going to be popular in school? I just watched the quarterbacks documentary on Netflix. Did you see this?
I didn't know. I'll have to check it out.
Well, anyway, like the point is like to people watching that, like the quarterback, NFL quarterback is the acme of human development, right? And it's like Kirk Cousins is Superman. And if they could, you're an MSU guy, right?
Yeah. I was there during Kirk Cousins, yeah.
Well, Kirk Cousins, I don't know how he was in college, but in the documentary, because he plays for the Vikings now, and he's not the top level for NFL, but he's a really good quarterback. But if you look at his life, how disciplined the guy is, the guy's like totally fit and ripped. He's like doing everything he can to be a better quarterback. And every weekend he engages in like a heroic competition, which uses his brain a lot, actually, as well as his athletic gifts. But like the average person can understand that. They can say like, wow, if you watch this documentary, you'll come out of it thinking like Patrick Mahomes and Kirk Cousins, Mariota, even these guys are the ideal. Their lives are heroic and exciting, and everybody can understand why they're awesome. Whereas, like, wow, I heard these two geeks sit around for an hour just talking about some math and like DNA or something. Like, what the hell is that all about? Why I wouldn't want my kid doing that, right? So that's my view of it.
Maybe it's like the great filter. You get like just smart enough to get gene editing, but then the preferences of everyone just shoots us towards like just completely hedonistic lifestyle and making everyone star athletes and just living for pleasure and fun.
Oh yeah, it could totally go that way. But the thing is that there are enough people who are smart and want smart kids, that they're going to shoot off in some direction. So let's imagine a world where, I don't know how big of a Dune fan you are, but let's suppose we have do you know what the Butlerian Jihad is?
Yeah, they like kill all the robots, right? Or
Yeah,
they get rid of technology.
yeah, in the Dune universe, the humans have a close brush with AI taking over, but they defeat the AIs. And then they pass this law on penalty of death: thou shalt not create a machine in the image of the human mind, i.e., no LLMs. Okay, so anyway, they managed to cut off AI research, and it's just illegal. So their technology base is very strange. They have some advanced stuff and some very primitive stuff, but that's the Dune world. Imagine that we have the Dune world, so we don't have to talk about machinic intelligence, but we continue developing genetic engineering somehow, and then what happens? So yeah, you might have a lot of people that all they care about is that their kid looks like Kirk Cousins or something, right? And then, but that sliver of people who are already like in the 1% of intelligence, that's a big enough group of people that they can conduct their own effort. And now what'll happen is, behind the scenes, instead of like AIs developing new tech that reshapes the world, it's these genetically engineered super brains that are developing the quantum computers or whatever it is that you're allowed to do in this hypothetical universe, that are actually secretly shaping society behind the scenes. But this bulk of people have no idea. Just as today, this bulk of people actually really have no idea, like, what are the forces shaping society? Like, what are these solar panels, and does it matter that the cheapest way to generate electricity now is actually from solar panels made in China? Like, what? Nobody in this bulk of the bell curve understands anything I just said, or what the consequences of it are, right? But it'll just be more of that on steroids in this hypothetical scenario.
Is there a lot of venture money going into this? Because it's sort of weird to me that you don't hear really anything about these sci-fi potentials. Like I said earlier, you heard about the Human Genome Project in like the 2000s, and then it's been dead. And honestly, even as someone who just reads the internet all the time, you kind of have to go to like your blog or Gwern's blog or actually read scientific papers to get this stuff. It's not like really posted all over. There's a couple of pop sci articles, but it's not that popular. Is there a lot of like venture money going into it? Because again, it doesn't seem like there's that like so much could go right here, and it seems like the probability is like quite high.
So it takes a certain kind of reasoning to follow the path that you just kind of outlined. And that kind of reasoning is most prevalent in the EA world, effective altruism world, rationalist world, where they're trying to follow the science, they have kind of open minds, and they are interested in human flourishing. And they like IQ points, right? So in that community, there are people really interested in this, and there are venture capitalists in that community who want to fund this stuff. But believe it or not, even among the pool of venture guys, there are normies. Like, oh, let's build the next e-commerce platform. There are normies, and then there are guys who are like, I like deep tech, edgy investments. And it's those guys who would invest in like genomic prediction typically and stuff like this. So, or they just see the appeal of, they know IVF is going to blow up because for human fertility is going to become more and more of a challenge as people get as women pursue high-powered careers, and people get older and older before they have kids. So they just see that as a business opportunity, but they're not fundamentally interested in this, like some people would call it like transhumanist future or something like this. So it's a whole mix. Not all venture guys are like really open to these ideas.
It's just like it surprises me a little bit because it's not really in your face, but it seems like it could be really big.
Yeah. Oh, I think it's one of these things that will be really big. Like, I would predict before too long, this 10% babies produced through IVF, which is true only in a few countries now, will become more than norm in developed countries around the world. So at that point, you're talking about 10% of all babies born. And then if like there's one company that actually can do embryo genotyping in that space. Wow, that's a pretty powerful company. It's got a lot of resources. Wow, they're actually assembling the data set for intelligence themselves on the down low. And wow, they're the only ones now that can actually predict IQ. And by the way, they can demonstrate there's an asymmetry related to this ML and polygenic traits, which is that I don't have to reveal my predictor to you in order to prove to you that my predictor works. So in other words, let's suppose I make the public claim, hey, you know what, genomic prediction, we finally, despite the attacks of all you woke morons, we've actually completed our cognitive ability predictor, and it correlates 0.6 with actual IQ, and it's good enough, we're going to start making superhumans, fuck off, right? So, and oh, by the way, the government of Singapore has invited us to build a 10,000 square meter, research institute and genotyping lab there. If you want to bomb Singapore, go for it, but otherwise leave us alone. Now, oh, and by the way, people from all East and South Asian countries now are flying to Singapore to have their kids to do their IVF cycle. Okay, suppose that's the case. Now, you're some normie computational biologist at Harvard funded by NIH or something, and you're like, Steve Hsu, I don't fucking believe you. You guys are lying. I don't think your predictor works. I would say the following. I'd say, okay, pull a few hundred people from people that you've genotyped, and you just happen to know their SAT score. You don't tell me their SAT score, but we're going to give it to a third party, Dan Schulz. He's going to hold the true SAT scores of these few hundred people, or 1,000 people, but you're going to give me their genomes. Okay, then I just run my algorithm. Oh, it took me 5 seconds on my iPhone. I just run it. Here's the flat file with a list of 1,000 predicted cognitive scores, and then Dan will he'll calculate the correlation between my predicted IQ score and the one that was actually in the biographical data of these 1,000 individuals. And Dan says, yeah, it correlates 0.62. And I would say, hey, Harvard guy, go fuck off. By the way, I'm not telling you what the structure of my predictor is, but it works. Give me another 1,000. I'll show you again. So we can get to a point where I can prove to the world I can do X without revealing how I do X. And, at that point, just to repeat myself, I'll just be like, go fuck off, guys. See you later. You don't like it? Fine. Hey, you in particular, when you want to do your IVF cycles, your money's no good here. We're not servicing you. Get the fuck out of here. So that, will we ever see that day? I don't know. Maybe the AIs will take over before any of that shit happens.
This raises a question though, because if this gets that good, is there a way to run a calculation? So for me, I just got married, right? Is there a way for me to run a calculation? Thank you. And my wife, she actually asked if I would ask you about this, but she has one of the variants for CHEK2, which is like one of the more rare breast cancer, puts you at elevated risk for breast cancer.
Yes.
And the question was, is there a way to run a calculation on her genome and my genome to see what the risk would be and if we could benefit from IVF? And presumably, like, IVF is a lot to go under, but at some threshold, it could make sense if you had a really high chance of passing it on. So pose the question to you.
Yeah, so let me make two observations here. Let's suppose in your family there is a rare genetic mutation. Either you or say your wife has this rare genetic mutation. And let's suppose that even one copy of it is dangerous. Okay? So you should then go through IVF because we can do the following. We do the following. We take saliva from mom. We take saliva from dad. The region where that CHEK2 variant is, we then know what her surrounding haplotype, the SNPs around that, what state she has there, what that chunk of DNA looks like for her, and what that chunk of DNA looks like for you. And we can then look into the embryos and say, these are the embryos that got the CHEK2 variant from your wife, and these are the ones that didn't. And you might consider, if you're worried about the effects of that, use these, not these. So that's just a Mendelian, what we call a Mendelian or single gene variant. And we can pretty much 100% guarantee you your kid is not going to have it. if you go through embryo genotyping. Okay, so does that help?
It does. What would the chances be if we didn't do it? You just do it naturally. Is it?
So I don't know about check 2 whether it's recessive. Like, does your wife have 2 copies or 1 copy?
I'd have to ask her. I don't know.
So she has 2 copies. Well, okay. There's some basically simple math that like any particular chunk of DNA, the embryo is going to get 1 chunk from mom and 1 chunk from dad, but she could get the chunk, like if your wife only has 1 copy, she could get the chunk of the DNA that doesn't have that variant from mom. So like 50-50 that she gets at least 1 copy of it from mom. But we can just like tell you which ones are which, and then you don't implant the ones that have the dangerous variant. So these Mendelian issues can be solved. That's like the easiest thing for us to do. I just want to make one, because you mentioned breast cancer. I think this is like as a public service announcement. I always make this point because I think people are unaware of this. So many people are aware of the BRCA mutation. So that's mutations on a particular gene or region that predispose women to breast cancer. Okay? And it's pretty rare, like only like, depending on your ancestry, your ethnic group, only like roughly kind of 1 in 1,000 or a few per 1,000 women have this genetic variant. If you do have it, if you have the worst version of it, you could have like a 60-70% chance of breast cancer, by the time you're 40 or whatever. And it's very, there's a broad awareness about these BRCA variants, even though in aggregate, the number of women or the number of families that they affect is really quite small. Okay. If you're doing polygenic analysis of breast cancer, what you find is that there is a contribution to breast cancer risk from these rare variants, which is only affecting a very small number of people. Then the rest of the population, most of the variation in whether you're high risk for breast cancer or low risk for breast cancer, comes from about 1,000 individual loci, which are each only adding or subtracting from the risk a little bit. It's a typical polygenic story. But most people who are high risk for breast cancer are high risk for the polygenic reason, because so few people actually carry the rare mutations. Even though a rare mutation is very dramatic to talk about, and that one little change has a huge impact, that's in a sense, negligible part of the population. What's really going on is, just like there are some tall people and there are some short people, there are some high breast cancer risk people and some low breast cancer risk people. And if you define the set of people who have as much risk as BRCA carriers for breast cancer, there are 10 times as many people who just happen to have the polygenic combinations that make them that at risk than there are actual BRCA carriers. Now, we have the technology to compute from your 23andMe genotype. I can compute your polygenic breast cancer score. So there are 10 times as many women walking around not knowing that they're high risk for breast cancer as the entire aggregate population of CHEK2, BRCA, blah, blah, blah carriers. But they don't know, and they're not told that they're high risk for breast cancer because people are just dumb. They're not, the medical profession has not updated on the new information coming from this science in polygenic risk. The fact that we can compute this, and the fact that the number of affected women defining some threshold is 10 times larger than the number of women walking around with BRCA or CHEK2, that just hasn't penetrated our medical system yet. And 10 years from now, I'm sure people will be getting polygenic scores for like colon cancer and breast cancer and all this other stuff. But currently, it hasn't penetrated. It hasn't, at the moment, penetrated at all into the general consciousness of doctors. Now, in our embryo practice, we often will see a family. They come in and they're like, you know what? We have a lot of breast cancer in our family. But, wow, we're BRCA negative. We're not carriers of any rare breast cancer thing. But my aunt had breast cancer and my grandma had breast cancer, and I'm really worried. Can you make sure that my baby girl has lower risk for breast cancer?
So now we get the genomes and we're looking. We get mom's genome, dad's genome, and the embryos. And we look and we say, yeah, that's right. This family has high, mom's part of the family, oh yeah, and even maybe dad's part of the family, has high polygenic risk for breast cancer. They are not carriers of the rare variant. They just happen to be unlucky. Just like this family, oh, a lot of tall people in this family. Oh, in this family, a lot of people who are going to get breast cancer. Then we look in the embryos. But the way that mom and dad's genomes recombine mean that even though the family in aggregate is way above average in their polygenic risk for breast cancer, we typically can find embryos that are at normal risk, just because of the luck of the draw of the way that the different, like they didn't get the risk variants in this chunk, they got the low risk chunk from dad, and they got the low risk chunk here. So that embryo is fine. But without our technology, there's no way. They're never going to break this chain of inherited family history of breast cancer. Whereas we can break that chain, we just break it right there. So that's like the most interesting stuff for me personally, to like look in to see like, oh, what's the spread of, what's the distribution of breast cancer risks of these embryos? And oh, what were mom and dad like? We can just see it. It's like amazing new technology that didn't exist, 5 years ago, but we can do stuff like that.
Well, presumably you all have like some of the best models for this stuff. Have you considered a consumer website similar to 23andMe where instead of screening for kids, you just like let people off the street run their polygenic scores?
We want to do that, and we're we every now and then some other entrepreneurs come to us and say like, hey, they have the insight that you just had, which is that somebody should be pushing this as an adult polygenic score product out in the same way that 23andMe or Ancestry do their thing. And we've really just been kind of heads down on the embryo stuff. And so our company has not tried to do this, but we have the back end for calculating everything. And the embryo report that we generate could easily be an adult report. So we could start this company. In fact, actually at this moment, it just happens like we just had another iteration of another set of entrepreneurs coming to us saying, hey, we want to pursue this adult polygenic score market. And so we're kind of talking to them. But so far, nobody has succeeded in doing this. Just because it's kind of complicated, and like people are kind of like generally concerned about their health, but how much do they really care? Whereas like you're very focused when you're going through IVF. It's like you decided, I really want to have kids. We're having a fertility problem. I'm going to invest tens of thousands of dollars. And oh, wait, I can genotype my embryos. Wait, let me figure out what that means. It really focuses the mind. And it's qualitatively different from like, you go see your GP at the hospital. And he's like, yeah, Dan, we checked your blood lipids. You're a little high on the, maybe, do you eat a lot of red meat?
Lots, yeah.
Yeah, and then you have that conversation with your doc, right? Now imagine that dude explaining to that dude polygenic scores, pleiotropy, machine learning, blah, 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 absolute risk versus relative risk, odds ratios. Just imagine explaining all that to that guy who has to see like one patient every 6 minutes or whatever the hell the HMO is doing to this guy. It is at least somewhat challenging, I think, to contemplate getting. It'll happen eventually, but for any particular entrepreneur to try to want to do it, I'd say it is a difficult lift.
Yeah, I've actually always been just surprised by like the general population, how many people just like don't seem to think 23andMe is that interesting. Like I find it like super insightful to like learn your high risk and stuff. A lot of people just don't care. It doesn't seem to be a big deal.
Don't care, and also you're an engineering graduate from Michigan State with a background in tech. Like, oh, are you how representative are you of the general population, right? So.
You have some like really interesting personal stories, and a lot of, for at least a couple degrees, some famous people that I think are interesting stories you post on your blog. The first is Jeff Bezos.
Yeah.
So you work with a lot of physicists who used to be students with him at Princeton, is that right?
So Jeff Bezos went to Princeton to study physics. This is actually in many interviews he's given, he discusses this. When he left high school, his goal was to become a theoretical physicist. He went to Princeton, which has one of the top undergraduate programs in physics. He did well as a physics major until he got to quantum mechanics. And at that point, he himself says he had a lot of trouble with the more abstract formulation of quantum mechanics, and he realized that out of his class of roughly 30 physics majors, or whatever the number was, physics majors taking, it would have probably been 20 to 30 or something, taking quantum mechanics with him, there were a couple that just got the material naturally, and he felt they were just much better than him at it, and he just felt he didn't have a good future as a physicist, and so he switched to computer science. So that's all on the record. My best friend from high school went to Princeton, started out as a physics major, switched to math. I'll dox him a little bit. He graduated #1 in his class, Bezos's class from Princeton, I think class of '86. My buddy was the valedictorian of the class of '86, and so was pretty good friends with Bezos. They were both in the same eating club. I think they were both in Cloister. And then, so I heard a lot of stories from him, and then there's a whole nother group of physics guys who, for some reason, it was like fashionable or whatever, for whatever reason, a lot of Princeton undergrads would go to Berkeley for their PhD in physics. And I also went to graduate school at Berkeley. And so I'm class of '86, so my high school friend was a year ahead of me, but I graduated in 3 years from Caltech. So we ended up graduating college the same year. But in my cohort at Berkeley in graduate school, including my roommates that I lived with, were guys who had taken quantum mechanics with Jeff Bezos and knew Jeff Bezos. So that's how I heard all these stories about Jeff Bezos. And anyway, so yes, so I kind of know these stories firsthand. Sometimes people will say things like, Bezos wasn't smart enough to be a theoretical physicist, but he was smart enough to be one of the greatest entrepreneurs of all time and business creators of all time. And to some extent, like to the extent that like general intelligence is a thing, I don't think that statement is completely inaccurate, because probably like all these other physics dudes from Princeton who were contemporaries of his were, in some like broad intellectual sense, like as sharp as Bezos. But of course in life they were nowhere near as successful as Bezos. So, whatever. But yeah, I mean, there are levels to this thing. My wife hates it when I say this, but there are levels to this thing. So like you could easily be like, if I said it the following way, like this guy was a great high school running back, but when he got to Michigan State, he could not make the starting lineup and eventually dropped out and became a rugby player or whatever. People would be like, oh, I get it. I understand that story, Steve. I'm not personally threatened by that story. Let's tell the story again. But if you tell the story about Bezos, there are a lot of people who don't like the story. But as far as I can tell, the story as I've related to you is true.
So basically he was in college and he realized that he was just like not up to chops with some of the other physicists. But then he goes to Amazon and he's like the smartest guy in the room in every conversation that he has.
Right. So this part of it I can't personally verify. This is from other people's accounts. So other people will say, and I guess the part of it I can verify is the experiences I've had as a generalist in tech startups, which aren't really physics-focused startups. But the statement is, there's somebody, because there are levels to this thing, you can have a guy who, is not an expert at a particular thing, but the engineers come in and start talking to him and saying, hey, this is how we decided to solve this problem. We're going to do this. This is our process. And the other guy, who could be Bezos or it could be me as CEO of one of my startups, I could be like, okay, I think you guys have done a good job, but did you think about this? Or did you think about this? Or the one part of your plan is not going to work is like, if conditional on this, that's going to not work, right? So all these stories are that, like, in many situations at Amazon, Bezos was the smartest guy in the room and was often very useful to have in technical discussions. People have said the same thing about Bill Gates, who started at Harvard as, wanting to study math. And I've experienced it. I'll just say I've experienced it myself. Like if you ask engineers at my companies, like if we get Steve in a technical discussion, he might not be as up to speed on the details as us, but he's often very useful to have in the conversation and sometimes says stuff to us that we didn't think of. So I think that's plausibly true.
The one other famous person that you had some interaction with Richard Feynman. So I had the Feynman lectures when I was like a freshman at Michigan State. I was like using those, and instead of whatever physics books they like gave you, my dad passed them down to me. But I guess my question here is like, he's from an era with some of these like legends, Bohr, Fermi, Einstein. Like you can go watch the Oppenheimer movie, right? And it's like filled with little like Easter eggs of all these guys, and they're honestly, even people who don't know science like know a lot of those names. And my question is like, where is that today? Is it gone because maybe it's still there? Maybe I'm just ignorant to it, but is sort of like the famous scientists gone because those ideas were easier to find and we got the low-hanging fruit? Or is there something different going on where we're just producing less of that massive talent and it's not all working together in the same room? Like what do you think is happening?
This is a great question, and it's something, even professional physicists talk to each other about. So let me make a couple observations. In the era pre-Feynman, the earlier generation would have been like Bohr, Oppenheimer was a little bit younger than Bohr, Heisenberg, Dirac, people like that. And then after that would be like Feynman, Schwinger, and some other people. At that time, if you just calculate how many people had the benefit of a university education like what you got at Michigan State? What is the number? It's literally a tenth the number that have that opportunity in the modern Western world. That's not even counting like developing countries, right? Like China, India, stuff like that. So you're talking about a much smaller pool of people from which you could draw top scientific talent. Another way of convincing yourself of that is go look at the raw scores of the Army IQ tests of people who entered the U.S. Army. They started using IQ tests for the U.S. Army around World War I. Okay, so they have records like 1918, 1925, There's a steady increase in the raw scores that's called the Flynn effect. It's not because people were genetically getting smarter. It's that, wow, let's just match it up to what was the average number of years of schooling that a World War I recruit in the U.S. Army had? 6. What was the average? 6. I mean, today that would be child abuse, right? That's literally the kid starts grade 1, leaves school, grade 6 to start working in the farm or the factory or something. So there was a big change in just the chances that you got a decent education between 1920s when these giants were around or getting educated and today. So we're talking about a much bigger pool of people from which to draw talent. Okay? Now, similarly in sports, if you're like, how fast did Jesse Owens run in 1936 to win the Berlin Olympic Games? I think he ran like a 10-3, 100 meters or something, right? And that was like no spikes. I was probably running on like dirt or something. Who knows? But anyway, in those days, like you could have like, here's the distribution of the other runners, and then here's Jesse Owens. You could have like a wild outlier. Who Jesse Owens, like by modern standards, maybe he'd be a pretty good sprinter. Maybe he'd be not quite world class. Maybe he would be world class. But the point is like he was an outlier in that pool of athletic talent. He looked like a giant. He looked like a superhuman compared to all these other guys because the other guys sucked, right? And similarly, like a guy who by modern standards would be like, this guy is a pretty competent, he's a world class but not considered super genius theoretical physicist. Back in those days, he might have been an outlier. He might have been a guy who looked very different from the bulk of the rest of the distribution. So when you're sampling from a smaller pool, it's easier to find outliers that look very different than everybody else. But when you have enough people, the distribution starts to fill in. And then person X is really smart, but person Y is just as smart as person X. He's right next to the guy in the distribution. Then there's another guy, person Z. And actually in this bin there are 10 guys. And in this bin, so then after a while you don't talk about person X is a genius outlier. You're like, person X is very smart, but I can identify 100 other people who are similarly smart. And they're all working on quantum gravity. So they don't stand out. There's two distinct issues here. Who has the ability to make a contribution, like pushing the science forward a bit, who stands out so much that they start to mythologize the guy and talk about how brilliant he was and, you know, how off scale he was and there was nobody else like him. So those are two different things. In the modern era, there are so many smart guys that, and our systems are actually not bad at picking them out, right? That, you know, there isn't that much overlooked talent, or a lot less overlooked talent. So anyway, the distribution kind of filled in, and so just psychologically, it just feels different. Now, the second thing you might say is, well, but how come we're not making these like huge leaps of progress that happened? There happened to be like huge leaps in like special relativity and quantum mechanics in the early part of the 20th century. That has much more to do with with factors outside our control. It just happens that the difference in energy scales between the weak scale, which is very well understood in the standard model of particle physics, there are about 10 to the 16th orders of magnitude between that scale and the quantum gravity scale. So we don't have any experimental equipment that can probe the Planck scale, the quantum gravity scale. So the theorists can talk all they want about what's going on here, and maybe string theory is like the most beautiful, correct theory ever developed, but we were not going to know it, certainly not in my lifetime or your lifetime. So the problem is actually there just happens to be a big gap in fundamental physics between what we've explored and what we need to explore to test the current set of theories that people are working on. In the old days, if you look at like what Lawrence, like if you saw the movie Oppenheimer, there's a character Lawrence. Lawrence was one of the great experimentalists of his day, and he built the first cyclotrons at Berkeley. And you may ask, like, well, how much money did that cost? How long did that take? How hard was that to build those cyclotrons? Because as soon as they built the cyclotrons, they could then test a bunch of interesting questions that the theorists were worried about. So in those days, like you could be like, theory is tested within 6 months, and then theories were formulated, and it was a very healthy, good time for physics. It was a golden era for physics. Whereas for fundamental physics, particle physics today, we have this problem that nature is not kind. For some reason, nature put the quantum gravity scale way beyond the scale at which, you know, even the biggest accelerators on Earth can explore. It could have been differently. There's some other parallel universe where the Planck scale is very close to the scale of like a 1 kilometer radius collider, what that can do. And in that universe, you know, Steve Shue won the Nobel Prize for like some theory of quantum, some theory that was some experimental prediction that was tested like when he was still 27 years old. But what can you do? We don't control how nature is structured. It is what it is. And some generations get lucky and the experiment and the theory are kind of close together, right? Like I could write the paper about, oh, height is going to be solved pretty soon as we get to here. And then, oh, a few years later, yeah, we solved height, right? So I could do that only because I was lucky to be in it. Well, I kind of purposely went there because I knew this was going to happen. But, you know, I was lucky to be in a place where the underlying technology to explore genomics was advancing very rapidly, and the theory could actually play a role in understanding what the experimental results were. But that's different from like, are there still smart guys? Are there still geniuses? Like Terry Tao is probably in some ways, like in terms of his mathematical power, way beyond some of these guys, like certainly way behind, like way beyond like Oppenheimer and Bohr and Heisenberg and those. There's no question. Like those guys are not even in the same league, not even in the same level as like a Terry Tao or something like that. There's just no question about that. So, anyway, I don't know if I helped explain.
No, that was that. Like sometimes I feel like the answer to that question is kind of fluffy, and people just like blame like, you know, school's not as good anymore. We don't do it like we used to. But that actually makes like perfect sense. It's a combo of the field that you're in. They happen to be in a field where you could like test your theories on short notice, and then also just you're going to stand out if there's only a couple thousand of you versus, you know, 10, 20, 30 more thousand.
Absolutely. I mean, the other factors that people quote, like, oh, they used to have this aristocratic education, and it is true. Like both Feynman and Van Noyen,
yes,
they had tutors. They came from wealthy families. They hired people that they were equivalent of PhD students to come and tutor their kids when they were still like 15 years old. And so, of course, you would learn a hell of a lot more if you didn't have to go to Okemos High School. I don't
Yeah.
know. This is Okemos High School, but instead, like, Dad brought home some grad students who started tutoring you in physics and computer science. Like, I think that might work better, but we don't do that anymore. So,
Yeah, there's that like Bloom's two sigma problem, right? You get like two standard deviations or something of improvement in education if you're like single tutored or whatever. But at the limit, that like shouldn't really matter because at some point you still got to go out on your own and like, you know, just do the physics. So
exactly,
that doesn't, yeah.
exactly. I mean, of course, it's as a modern who grew up with this stuff, we're a little bit biased because when we look back and we're like, wow, what passed for genius work in 1929, it's like, wow, this is pretty simple stuff. Like, this is stuff undergrads can do now. And of course, I don't mean like undergrads know it because it's already in the textbooks, but just in terms of like the difficulty of the calculations that those guys had to do, it's like, it's not really that impressive by modern standards. So, you know,
So should more physicists be jumping fields? I mean, that, you guys do it a lot actually right now. I feel like physics majors are all over the place in other fields.
yeah.
But should more be hopping? Is it too tough going, or what's your take on that?
I think that it's all a question of personal taste. So, like, if I look back, like, I'm an old guy now. So if I look back and I say, like, wow, what did I do in my life? Well, how did that happen so fast? Like, what did I actually do? I take great satisfaction in those years of closing the gap between being a young kid learning physics and getting to the frontier, like to understand everything between like the elementary stuff and the frontier, pushing the frontier at least a little bit forward in my own way, but developing a full mastery of everything in between. And that does take a long time. I don't think anybody can really just do that in a few years. Even like when you go through the PhD program, you still don't really have a full, you know, maybe one area you've closed that gap. But across a broad spectrum of physics to really do that, the intellectual achievement takes decades to do. Now, and you asked me like, well, Steve, if you had just abandoned that a lot earlier, your net worth there, you could add like another zero or two to your net worth. What's the trade-off, right? And different people are going to react differently to that, right? I don't know what to say. Like, I kind of feel okay. Like, I feel like my satisfaction with having mastered these concepts in mathematics and physics and biology and computation is very valuable to me internally, even if it didn't return more than like a professor's salary during that time interval, right? So it's all a question of personal judgment. If you know ahead of time that like, hey, fundamental physics is not probably going to make a lot of progress in the next few decades, but AI is going to like do this, I would say like, hey, you got to have, unless you have a very, very strong affection for fundamental physics, it will probably be more exciting for you working on AI, you know, but I can't make that decision for somebody else. Somebody else has to like have their own, based on their own preferences, make that decision.
Some great words of wisdom to end on, Steve. You've been super generous with your time. I've had just a total blast talking with you. Thank you so much for coming on.
Yeah, this has been a great conversation. Good luck with your podcast.
Thank you.